Okay, you have you guys have seen this before. You know what we are talking about again, but we are going to talk about it a little bit more in detail at the beginning of semester. The material for first exam. What is the name of that element? It is not element, but that group. It's a phosphate. You remember that? This is a phosphate, right? What is that one? What does it look like? Lab practical exam, lecture exam question. What does this look like? It's a what? It's a sugar molecule. It's a wrong sugar molecule, you said. That's a sugar molecule. That sugar molecule is ribose. Right? And then what is that one? Phosphate again. What is that one? The sugar molecule. Ribose. Okay, what is that one? Phosphate. What is that one? Again, the sugar molecule ribose. What is that one? Phosphate. And what is that one? The sugar molecule. Now it is called deoxyribose. It means it is because this number 2 is missing one oxygen. That's what they call it, deoxyribose. We have another molecule called RNA, which that one has oxygen attached to it. It's called ribose. This is D in front of a word, it means without. Right? Am I making some sense? So deoxy, it means without oxygen, and of course ribose is the pentagon, 5-sided, 1, 2, 3, 4, 5. Another, that you don't get scared, another quick question. What is this element? Oxygen, very good. And what would that be in that corner? They didn't draw it. Carbon, very good. What would that be in that corner? Carbon, 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 and of course they did draw carbon here. Now, my question is, between that oxygen and that carbon, there is a dashed line. What does that line indicate? It's what? A single what bond? Covalent bond. Good. The quiz is not a teamwork today. Right? If it was a teamwork, you all would get 15-10. It's not, you all get 10, but it's not. You have to individual quizzes. Is that right? In this individually. Yeah, it's a covalent bond. This is a covalent bond also, that line. This line is also covalent bond. That line is covalent bond. That line is covalent bond. This line is covalent bond. Covalent bond. Am I making some sense a little bit? So the structure of DNA, what they discovered, Watson and Crick, they said it is a ladder, look like a ladder. Okay, the steps, not the steps, the legs of the ladder. The legs of the ladder is phosphate, sugar. Phosphate, is that right? Sugar. Phosphate, sugar. Phosphate, sugar. That's it. Phosphate, sugar. Phosphate, sugar. The legs. Phosphate, sugar, phosphate, sugar, phosphate. I hope I'm making some sense. And then, it's not over yet. Those are the legs of the ladder. They have to have steps. The steps of the ladder are your what? That was your lab practical exam question also. What are these? Generally, what are they called? Huh? They're called nitrogenous bases. In the lab practical exam, I put it out for you, the picture of it. I said, what is the name of the molecule? You didn't have to write down thymine, adenine, cytosine, guanine. You wrote down nitrogenous bases, and you got credit for it. Am I making some sense? I did draw these for you. Nitrogenous bases, this is what? What element is that one? Nitrogen, nitrogen, oxygen. How many covalent bonds between this oxygen and that carbon? 2. Right? Between that nitrogen and that carbon is covalent bond. Between that nitrogen Between that carbon and that carbon, there are two covalent bonds. Am I making some sense, everybody? I don't expect you to memorize that thymine look like this. Of course, you should know thymine has one ring. Adenine has two rings. Adenine is a purine. Thymine is a pyrimidine. Pyrimidine, it means it has one ring. And purine has two rings. You remember this information from first exam? Material from first exam? Okay. So, what they proposed, the legs of the ladder are attached to what? To sugar or phosphate? The nitrogenous bases are attached to the sugar or to the phosphate? Sugar. So, right here is one nitrogenous base. I'm going to write down T. And the other sugar molecule right here attached to it, A. This one, sugar molecule, guanine, or cytosine, and this one, right here, again, guanine. Am I making some sense? Now the legs of the ladder are your nitrogenous bases, always. Do I make sense? So the legs of the ladder are sugar phosphate, sugar phosphate, sugar phosphate. The steps of the ladder which you climb up is nitrogenous bases. Nitrogenous bases, they pair up, one from this side and one from that side. Always. It's scary in biology, you use the term always. But this is one of the few times I use the term always in this class. One of the few times. Always. 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 Thymine binds to adenine. Thymine binds to adenine. And guanine binds to cytosine. Guanine, cytosine binds to guanine, and adenine binds to thymine. Do I make sense so far? That's the structure of DNA. That's one of the essay questions. What is the structure of DNA? We are not there yet. We will be. We will be there. Have you heard of that saying? Do not walk in front of me. I may not follow you. Do not walk behind me. I may mislead you. Walk by me and be my friend. You've heard of that saying? Okay, so hang on. Stay with me. Okay, so right here, animation. Here, of course, this is not straight ladder, it's a twisted ladder. Have you seen those fancy houses?
Yeah.
And they have twisted staircase. Next time you go to a fancy house and you see twisted staircase, they go down, oh, that's DNA. Impress your date, or your husband, your wife. Your date is wrong. Impress your wife, your husband.
DNA and RNA are nucleic acids. Polymers
We
made of
should not talk about
subunits
RNA
called
here
nucleotides.
now, but that's
One difference between DNA and RNA is the type of sugar their nucleotides contain. DNA contains the sugar deoxyribose, while RNA contains the sugar ribose. Ribose has one more oxygen
Are you
atom
watching?
than deoxyribose. DNA and RNA are each composed of 4 different nucleotides, which differ in their nitrogenous bases. 3 of the 4 bases are the same in DNA and RNA: adenine, guanine, and cytosine. The 4th base in DNA is thymine. In RNA, it is uracil. The nitrogenous bases guanine and adenine each have 2 linked rings of atoms. They are called purines. Cytosine, thymine, and uracil each have a single ring, and these 3 bases are called pyrimidines. For convenience, the carbon atoms in a nucleotide sugars are numbered, beginning with the carbon atom bonded to the nitrogenous
very
base,
important. I
moving
forgot to
around
mention
the
that.
ring, and up to the carbon that is bonded to the phosphate group. The 1' carbon is bonded to the nitrogenous base, the 3' carbon to the next nucleotide, and the 5' carbon to the phosphate group. DNA and RNA are polynucleotides, long chains of nucleotides. Polynucleotides are always assembled in the 5' to 3' direction. A covalent bond forms between the carbon at the 3' position of a nucleotide and the phosphate group at the 5' position of the next nucleotide.
Okay, something I did not mention.
RNA usually consists of a single polynucleotide chain. DNA, on the other hand, consists of 2 polynucleotide chains. The 2 DNA chains, or strands, are oriented in opposite directions and held together by hydrogen bonds between the nitrogenous bases on opposite strands. Because of their sizes, shapes, and arrangement of polar groups, the DNA bases form complementary pairs. Adenine pairs with thymine, and cytosine pairs with guanine. The 2 polynucleotides in DNA wind around each other to form the familiar double helix. The bases can be in any order, like the letters of the alphabet. However, the base sequence is significant. Sequences of bases called genes encode the instructions for the structure and function of an organism.
Okay. Now, answer my question. You watched the video, right? Are you ready for my question? Can you see? Why did I draw 2 lines here and 3 lines here? 3 lines, 2 lines. Why?
Sir,
Huh?
a double and triple bond.
Of what? Hydrogen bond. I'm glad. What? Maybe you should take the quiz today as a group.
We need each other.
Very good. That's it. Very good. You make a good team. 2 hydrogen bonds between thymine and adenine, and 3 hydrogen bonds. Hydrogen bonds from exam number 1 is the attraction, right? Of course, the molecules have to have hydrogen. They didn't. It's not in here. You will see it. They have to have 3 hydrogen bonds. 3 hydrogen bonds, 2 hydrogen bonds. So, can I ask you a quick question? Which one is stronger? Is this bond between cytosine and guanine stronger, or that is stronger between thymine and adenine?
It's a
Trick
trick question.
question? No, I never asked trick question. I had this friend in Minnesota. I said, Amir, anytime you hear questions like that, understand it's a trick question, and so give the other answer. But no, not me. I'm straightforward type of guy. Yeah, guanine and cytosine is a stronger bond because you have 3. If you want to break that, you have to spend more energy. You have 3 of them. Here you have 2 hydrogen bonds. This all will come into picture later on during semester when I talk about breaking these. It's easier to break those is easier than breaking these, and you will see that later on during semester. Okay, let's go. Rosalind Franklin, she took X-ray picture of DNA, and she showed this picture to Watson and Crick, and Watson and Crick took that picture. Their mind was prepared, and they came up with this. And of course, Watson and Crick won the Nobel Prize. Their name is in every textbook, and Rosalind, well, her name is in every textbook, but she didn't win the Nobel Prize. And of course, she died young because she was exposed to a lot of X-ray. Okay, she was exposed to a lot of X-ray, and I think that happens in history of science sometimes. So I said it once, prepare mind. You have to prepare your mind to take the advantage of the opportunity in life. Okay, so Watson and Crick took advantage of this X-ray picture, and they came up with the structure of DNA. But anyway, Franklin's X-ray crystallographic images of DNA enabled Watson and Crick deduced that DNA was helical. That's what it means, helical. It means twisted, like a helix. Okay, it's a twisted shape. That's what helical means. X-ray images also enabled Watson and Crick to deduce the width of DNA helix and spacing of the nitrogenous bases. The pattern in the photo suggested that DNA molecules was made up of 2 strands forming a helix molecule. So until then, everybody knew that DNA is responsible for inheritance, but they did not know the shape of it, how it is, and so on and so forth. They knew there's adenine and thymine in there, guanine and cytosine, sugar phosphate. They knew that, but how is it arranged? Nobody knew. Okay, the arrangement of it, they did not know. And that was the race. It was a race everybody was chasing. Here it is. Again, I should maybe, this is called anti-parallel. Let me go back. I forgot to mention this one. Where are you? Right here. Is everybody here? Is everybody with me? This is important for rest of semester. Okay. So the carbon here is called 1 prime. The carbon here is called number 2 prime. On the sugar molecule, this is the sugar molecule, ribose, right? On the ribose. 2 prime, 3 prime, 4 prime, 5 prime. Okay? So all of these sugar molecules called 1 prime, 2 prime, 3 prime, 4 prime, 5 prime, it has 5 carbon. This is a pentagon, it has 5 carbon. And this 5 prime is attached to the phosphate. And then here, there's nothing else attached to, it's a 3 prime end. So what he said in the video, that's a 5 prime end, and this is a 3 prime end. Am I making some sense? Do you remember protein molecules? I said always one end of a protein molecule is acid, the other end of protein molecule is the ammonia. You remember that? Same as DNA. DNA, one end of it is always 5 prime, starts at 5 prime and ends at 3 prime. And when new DNA is being building up, it's building up from 5 prime to 3 prime end. I hope I'm making some sense.
Yeah.
So the beginning, if you want to think of it that way, the beginning is a 5 prime and end of it. And Amir, where did you get 5 prime? Right here, sugar number 5. Here is sugar number 3. Okay, so sugar number 3 attaches the phosphate right here, and then 5 prime, 3 prime, 5 prime. And then of course the nitrogenous bases are attached to the 1 prime. Okay, so having said that, then what happens, the 2 DNA, these 2 strands are opposite of each other. One goes this way, the other one goes that way. That's what the idea of anti-parallel. This is parallel, right? My arms are parallel. Am I making some sense? But how about my arms now? They are anti-parallel. Right now my arms are parallel. Right now my arms are anti-parallel. Yes or no? Please.
This kind of confused me because
What?
they're still parallel, right?
They are parallel. My arms are parallel, right?
Yeah.
But are they parallel, or they're anti? I know it's confusing. They're parallel, but they're anti each other. The arms are
Oh,
running.
they're going the other way.
You're right. Look at my arms. This one, my fingers is this here way, is going this way.
Okay.
This one, my fingers are going this way. They are parallel. You're right, but they're anti each other.
Yeah, they're going opposite.
They're going opposite direction.
Okay.
How about now? My arms are going in same direction. They're parallel. Am I making some sense? That's the structure of DNA. DNA go opposite of each other. So one strand, the legs of the ladder that I draw for you guys, the legs of the ladder are opposite of each other. Again, how do you know, Amir, what do you mean by opposite of each other? 5 prime, 3 prime. This one, 3 prime, 5 prime. You remember this? If you forget it, think of my arms. They're parallel, but they're going opposite of direction of each other. My arms are going. How about now? My arms are going same direction. Are you guys do karate? Any of you do karate? You go like this. Oh, not karate. What is the slow motion one? Tai Chi. Thank you. You guys do Tai Chi. That's parallel, right? My arms are going parallel. But how about now? My arms are going parallel but anti. Yeah, still going parallel but anti. That's why you should do Tai Chi. Then you would understand the structure of DNA better.
You said that the video that the parallel lines go, like for example with that, the O is on the top and then the right, the O is on the bottom. The oxygen. So isn't it going the opposite way?
Yeah, up right here. The same as video. Same thing that the video said I said here. Yeah, the O is in opposite way. What O are you, what?
With the oxygen, or I don't know, it's like
Oxygen, yeah, right here. This is your oxygen. The sugar molecule is upside down. The sugar, if you want to look at it that way, the sugar molecule is up, right side up.
They're the same ones, they're just backwards. They're just going the other way. So if you were to take the 3 to 5 prime one and then put it on the other side, it'd go TGCA.
why is it the 3 prime? Is it because it's deoxyribose and then those in the middle are ribose?
Okay, reverse. 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. Then there's no more DNA here, right? There's nothing here. So which one is that? That's a 3 prime end. When I started, that's a 5 prime end, which a phosphate attached to it. There's no phosphate here anymore. If there was a phosphate here, then there should be another setup, another nucleotide here. And then on this one, this end, if you're starting from that end, this is a 3 prime end. There's no more DNA up there. And then on the bottom, there's no more DNA up here, but the sugar molecule, there's a phosphate here, if you want to think about it that way. There's a phosphate here, there's no phosphate on this end, on the bottom. Look at it, enjoy it. People worked very hard, people died. Rosalind Franklin, she died to figure out this. You know, Einstein, he was very thankful. You know all Einstein, huh? Einstein said, I'm thankful, on one of his speeches, I'm thankful for all of the scientists before me. They discovered their stuff so I can continue on. So she died. Think about it. All of that X-ray she was exposed to, she died young for discovering the structure of DNA. So it's our responsibility to carry it on. Okay? So anyhow, there's still a lot needs to be discovered about DNA, and not DNA, but how to manipulate it, how to play with it.
DNA consists of 2 complementary strands of nucleotides twisted together to form a double helix. In the space-filling model, each atom is represented by a small sphere. The yellow and red phosphate groups make the 2 sugar phosphate backbones easy to spot. They're on the outside of the double helix. We will highlight the backbones in blue. Now let's look at the crossbars connecting the backbones. Each crossbar is a hydrogen-bonded pair of bases, one from each DNA strand. An adenine is always paired with a thymine and a guanine with a cytosine. The double helix can also be represented by a ball and stick model. The
Ball and
simplicity of
stick, that's
ribbon
what you
models
have here.
often used, with ribbons for the sugar phosphate backbones.
Okay. Here is a here we go. That's the one. You see the 2 hydrogen bonds? You see 3 hydrogen bonds? You see 3 hydrogen bonds, and you see 2 hydrogen bonds. It doesn't matter. It does not make any difference. It's G on that side. That's what this is all about. Look, on one side is T, and one side is A. This side has A. This side has T. There is no law. It says, oh, this side have to have all T's, or this side have to have all A's. No. This side has to have all G's. This side has to have all C's. No. There's no law like that. But the law of base pairing, the law of base pairing says that always A binds to T and G to C. You don't like it? T binds to A, C binds to G. Am I making some sense? Yes? No? Okay, another animation.
DNA consists of two
Oh,
complementary...
we already seen this. I'm sorry. Okay, right here. So purine, purine 2-wide, pyrimidine. This is what Watson and Crick was proposing. What happened with Watson and Crick? He said both sides are what that is saying. I should have eliminated that one. They were proposing that A to A, T to T, G to G, C to C, but they saw that is wrong. That will not match the X-ray that Rosalind gave up. So that is not true. And they came up looking at the X-ray and being able to, they came up with that. Okay. Anyhow, right here is the 3 nitrogenous bases, the 2 hydrogen bonds, and then the 3 hydrogen bonds. Okay, basic principles, base pairing to a template strand. Since the 2 strands of DNA are complementary to each other, okay, each strand acts as a template for building a new strand. Okay, that's the next thing we're going to tackle. You remember in meiosis, it's a good thing that we already talked about meiosis and mitosis. In meiosis and mitosis, one DNA makes up chromosomes. Is that right? And protein molecules. Then you had to have 2 of them. That's the next thing I'm going to talk about. Not today, but on Monday when we come back. But I'm going to give you the prelude to it. I'm going to start talking about it, but not getting to it. Very seldom. Okay, so one DNA. How does one DNA becomes 2? That's what this is. Okay, so in DNA replication, that's what it means. One DNA becomes 2. One chromosome becomes 2 chromosomes. You remember? You remember one chromosome becomes 2 sister chromatids? Of course, the DNA has to duplicate. This is made up of DNA and protein molecule. This is made up of DNA protein molecule, so it's made. That's what the next we're going to talk about. The parent molecules unwind. Of course, the DNA has to unwind, become like this. That's the first step. And then unwinds, the 2 new daughter strands are built based on base pairing rules. And then Watson and Crick, they use a semi-conservative model. Semi-conservative model, it means that when DNA breaks, when this DNA breaks, on one side you have A, G, T, C, that's the old one, right? Right? And the new one is T, C, A, G. The old one is T, C, A, G, and the new one is A, G, T, C. Am I making some sense? New. And that's what it's called semi-conservative. That's what the term they tossed. They tossed semi-conservative. Half of DNA is the old one. The other half is the new one. This half is new one. This half is the old one. Okay, replication preceded when double helix duplicates. Each two other molecules will have one of the strands derived from the conserved from the parent molecule, and the newly one is made up. Right here is a parental molecule, which I did draw it for you guys, and now this is the new one, old one, new one, old one. Okay, then they use different experiments, and I will talk about this experiment later on a little bit on Monday. On Monday we'll get into the experiment. Is there any question? I'm done. Does anybody have any question about these things? You guys, you need to study these things. So when you come in here, we can discuss, we can talk about them back and forth. I showed you email, best email. Breakdance, did I show it to you? Marisa, please, you keep track of things, these guys. Bricks, did I show you the bricks? The guy put the bricks.
Yeah.
Earth and other planets, you saw that one. Gentle reminder. Brian. Two frogs. Did you see this? Have you guys read the story? No? Catherine? No? Maria? No? Two frogs fell into a deep cream bowl. Avita, are you ready? The one was wise and cheery soul. The other took a gloomy view. The other frog. Two frogs went to a bowl. Catherine, is that right? One was cheery. Yippee! Let's conquer the world. Let's carry on the DNA more. Rosalind Franklin died because of me. Is that right? She worked very hard. She was